Ko manaia te maunga, ko whāngarei te ringa parawa te moana, ko poki kauri te awa, ko takahiwai te whenua, ko takahiwai te marae, ko patuharakeke te hapu, ko Ngāti wai o rawa ko Ngāpui o ku iwi, ko Julianne Cheetham tōku ingoa. Our team is the Patuharakeke Tiwi Trust Board has a tile unit, and they've been sort of functioning for several years now, and they've been undertaking quite a lot of work up at Puke, which is the whenua up on the ranges, Takahiwai ranges behind our kāinga, behind our marae. It's a really significant place for us because our rangiora pā is up there and other important pā as well. And the land is regenerating kauri forests. There's thousands and thousands of kauri up there. It's some pretty healthy bush up there. You would have heard in my pepeha, I mentioned Puke Kauri Te Awa. Puke Kauri is a water body up the top there in the ranges, which actually is the source of our Takahiwai awa, which flows down into the harbour. It's a really central part of our identity, and one of the things I've been always sort of saying since being involved in the kauri kaupapa with this disease is that, you know, it's a threat to our identity. You know, when we say our pepeha, when we talk about these significant places, how can you have a puke kauri that doesn't have kauri on it? It's a real, you know, it's a real risk to our narrative, our kōrero, all those things that we pass on to our tamariki and mokopuna. From the beginning, we've been really interested in finding out all we can about this disease, this sickness, mate of our kauri, and how we can protect this sort of last place. And we know that we've got this disease in other parts of our rohe, in the Waipu end, over the back of Ruakaka on the other Kukunui side. But we were always pretty sure that puke kauri was clear or clean until a couple of years ago when we did have some samples in 2019 that came back. Just a small handful of around a couple of trees, and then we were really, really concerned because we thought we might have a new infection in there. But then our more recent testing hasn't been able to re-isolate the PA. So we're kind of in a situation now where we may or may not have kauri dieback up in the puke kauri blocks, and we really, really want to know one way or the other, because that will affect, you know, how we manage and make decisions going forward up there with all of our kaitiaki practices and things, not only that the tile unit does, but also that other whānau, you know, are doing up there. So you will have heard a lot about the disease triangle, and that is basically what this research is doing. It's looking at the host, Ngā Rākau, and the pathogen, Phytophthora agathodicida, and also the environment around it. So it's a whole of ngāhere approach, which is really coalesces really, really well with our Te Ao Māori thinking. We don't want to just focus on what the disease is doing or unhealthy trees. We want to look at what makes the ngāhere healthy or unhealthy, and how is the pathogen moving through our ngāhere or not.
So we're trying to map at the landscape scale areas that are more likely to show sign of decline in the canopy for the kauris, or symptoms for myrtle rust. And the way we do that, we're currently working with Ngaris in our team to identify some environmental variables that favor the expression of the disease. So whether it's areas that are downslope a track, for example, or downslope trapping lines, areas that are a bit more wet than others. We use like that different factors that we try to include in the model, and then the modeling helps you identify those areas that are more at risk than others.
So it's looking at what the soil's like Are there other types of phytophthora present that might be impacting the trees as well? And what else is happening in the catchment water-wise? What's the topography like? Is there a lot of pests and weed and just other health factors that are impacting the health of that ngāhere and causing that disease to get a hold and sort of start spreading?
So I think the idea behind all this is once you have this vision, you could, based on this, what we call expert knowledge, we could say in that landscape, this area is most likely to be infected than this one. So this is maybe where we want to concentrate our efforts to identify the disease front and maybe protect those areas that hasn't been infected yet.
You know, it's quite difficult because you don't always have symptomatic trees. You don't know exactly where the pathogen is in the soil and where it's moving, you know, down a catchment or in what part of the ngāhere and how fast is it moving. And those are the kind of things you need to know if you want to make management decisions. And that's also the kind of thing that mana whenua really want to know when they're trying to look at what they can do in their own forests to protect kauri.
So we've been involved with Pathogen, the team at Pathogen. We had meetings and trying to identify the specific trees that they would go and sample within their forest. So there was a process there where initially there was some identification of emergent kauri trees from the canopy, and that's done through aerial flights. And then once we had all the data on the trees that were in the Takahiwai forest, we had to, you know, obviously not every tree can go and be sampled. It would be great, but there is not the resources. So we randomly selected a group of those trees in conjunction with Julianne and her team.
When we were doing the site selection for this with Ngāri and the team, and also some of Waitangi and Dean's team, we were looking at, there was sort of, we were doing randomised sampling, but then we were doing selective sampling over that. So we were saying, well, what's on our rangiora pā do we want to look at? Because we looked at our rangatira rākau there in that area before, so we know we want to go back there. Are there any sites that have been randomly selected but fall into those areas, because those are ones we want to focus on? Or we might look at different whānau blocks and think it's all well and good doing, you know, remote sensing and getting the data, but we actually know from on the ground, well, it's going to be really nigh on impossible to get up to the back of that block because of the terrain, or we know that we have whānau or neighbours that are not going to want us to go up through their property. So applying all of that local knowledge has been part of the process as well.
So we went through that process with them and created some maps that they could then go into the field and, you know, find the tree and organize their field day, I guess the day in the field, and which group of trees they could go and sample. And from those trees they did some visual assessments and some soil sample. A group of them did soil sample, and the soil sample goes back to the lab with Ngāri. All the data that is collected using this survey form, that whatever gets collected, then as soon as the field day is finished and you've collected that data, the data goes back to a central, you know, data server, which we're not yet using the IIP because it's still in development, but ideally the process or the workflow is going to be that any field data collected is going to be stored straight into the IIP.
That's why our team, our Taio team, are really wanting to be at the forefront of the Research Area 3 mahi. We've had some training from Biosense We've got new gears, so we've got new tablets, new GPSs, all the gear to make sure that we can do sampling and surveying that's consistent with the approach that's been taken in the Waitakares. So the methodology that's being used is really consistent, and it's going to be applied in Te Roroa and Waipoua, Ngāti Rehia, Ngāti Rua, Te Uri Ahau and Tauranga Moana. So there's quite a few mana whenua that will be utilising this approach. I think our young fellas have been really good in tweaking the survey methodology, what works on the form and practice in the ngahere, which is going to be put, I think, is going to put all the other BMAs that are going to be starting this work very soon in good stead, because we've almost been like the guinea pigs or the pilot, sort of seeing what's doable on the new tablets. How does it all sort of come together? How many trees can you sort of sample in a day? And we're sort of working out what the work plan is and how much capacity and capability is needed. But we have been here for a long time. Our marae at Takihiwa is the only marae between Nguru and Omaha marae. And we have been really active in an environmental mahi since forever. What's good about the mahi up at Pukekohe, there's a mamae sitting under it, so it's about how do we get this place back, but at the same time it is kind of more proactive, positive work in terms of our tai tamariki and our team going up there and being able to have a look at what's in the waterways. We've had 2 wānanga up there where we've had our weavers from Patuharakeke and other whanaunga from around different rohe come to do kuta workshops and things like that up there. So we're trying to revitalise those practices, other rongoa, rāranga, those kind of activities. And this kauri dieback, kauri epidemiology research really feeds into this whole interconnected sort of kaupapa. How do we reconnect with this place, and what kind of practices do we want to do up here in the ngahere? Everyone's really into it, like not just our young fellas that are going up and doing the mahi. We take a lot of our kura kids up there and show them what we're doing up there. But it's also our koros and nannies and that are really keen too, because they don't get to go up there that much, and they remember the stories about what it was like up there. There used to be waterfalls up there and things like that are gone now because of the dam. People used to go up there to get pigeons and all of those kind of things. So we're trying to collect all that traditional kōrero as well as part of this and other projects we're doing. So it all ties into real kaitiakitanga, what we're trying to revitalise and achieve up there.